After that introduction, I'm actually a little bit scared. I think that was the nicest introduction I've ever had. I feel like I should just yield my time to David. But I'm grateful for the invitation to interface with this amazing community. It's warm and wonderful, and everyone's engaged and large. So thank you for making my first OTS experience such a pleasant one and such a stimulating one. I also have to begin by apologizing for my voice. I got COVID for the first time a couple weeks ago. And while I've been both antigen negative and PCR negative for many days now, my voice still sounds like Darth Vader's. So I'm sorry about that. All right, here are my disclosures, the most relevant of which is that I'm a co-founder of Beam Therapeutics and Prime Medicine. These are companies developing base editing and prime editing therapeutics. In this lecture, I'll overview the three major ways to edit DNA in mammalian cells, how they are being used to treat patients, and what bottlenecks remain. Gene editing is ultimately targeted chemistry on DNA. Nucleases catalyze the cleavage of phosphodiester bonds in the DNA backbone. Base editors catalyze the deamination of exocyclic amines of C or A. And prime editors catalyze DNA polymerization of specified sequences onto the 3' end of nicked target DNA. And many applications and variations of these three chemical themes have been described. These technologies are of major current interest because mutations in our genome cause thousands of genetic diseases that this community knows very well, that together afflict hundreds of millions of people. So a longstanding goal of the life sciences has been for many decades to develop the ability to install, correct, or otherwise modify all types of pathogenic mutations so that we can study or treat the broadest possible range of the resulting diseases. And thanks to the pioneering work of many remarkable scientists, some of whom are listed at the bottom of this slide, programmable nucleases, such as zinc finger nucleases, TALENs, and of course CRISPR-Cas9, really initiated the modern era of gene editing by enabling targeted gene disruption or deletion. Programmable nucleases excel at disrupting genes, which is, after all, exactly what nature evolved CRISPR-Cas9 to do, because the primary response to cutting the DNA double helix in many types of cells is to initiate end-joining repair processes that rejoin the broken ends of the cut chromosome. Now, most of the time, maybe 90% of the time, these end-joining events perfectly repair the cut DNA, regenerating the starting sequence. But a perfectly repaired DNA sequence, of course, can just be cut again by the same nuclease because it's identical, after all, to the target sequence. So this process of cutting and end joining occurs over and over again until the end joining makes a mistake that results in the deletion or insertion of nucleotides that prevent the altered sequence from being recognized again by the nuclease. The result are indels, these mixtures of insertions and deletions that unfortunately we can't control, but they do usually disrupt the function of a coding or a regulatory sequence. Now, disrupting genes can be very useful, both for basic science research and for certain therapeutic applications. For example, Cas9 nuclease has been used in a clinical trial to disrupt the gene that makes too much of a protein that causes transthyretin amyloidosis, ATTR. And when you disrupt that gene, you can observe a sharp decrease in serum levels of the protein in patients. And more than 200 patients to date, including Victoria and Patrick, pictured here, have been treated in clinical trials with CRISPR nucleases targeting DNA sequences that, when disrupted, offer clinical benefit. Most genetic diseases, however, arise from mutations that have caused genes to lose their function and therefore can't be rescued by further disrupting that gene. In other words, most disease-causing mutations really require precise gene correction rather than gene disruption in order to benefit patients. And using programmable nucleases to precisely correct mutated genes in most therapeutic settings remains very challenging. Nucleases by themselves, of course, simply cleave phosphodiester bonds and don't have any ability or even the information by themselves to make a specified change in a DNA sequence. In the presence of a donor DNA template, foundational work by Maria Jason and other scientists established that double-stranded DNA breaks can stimulate homology-directed repair, or HDR, which can correct pathogenic alleles by incorporating DNA sequences provided exogenously in donor DNA templates to replace the DNA sequences around the cut site. Unfortunately, the inefficiency of HDR in most cell types, as well as real challenges associated with making double-stranded breaks and delivering donor DNA templates, have thus far really limited the therapeutic relevance of HDR. We now know that double-stranded breaks induce undesired responses such as chromosomal translocations, large deletions, p53 activation. And as a result, if you look at all of the clinical trials of gene editing nucleases, they are thus far overwhelmingly limited to those indications for which gene disruption or deletion can be therapeutic, rather than the much larger set of indications that, in theory, could be addressed by precise gene correction. So to enable efficient and precise gene correction in a wide variety of cell types, we developed base editing and prime editing. Base editors and prime editors don't exist in nature, at least we don't know of any examples of them. Instead, they were engineered and evolved in our laboratory. Base editors are shown in this slide. They use the targeting mechanism of programmable DNA-binding proteins, such as disabled Cas9 or tail or zinc finger arrays, to engage a desired DNA sequence of your choosing. But instead of cutting the DNA, they use deaminase enzymes, either taken from nature or evolved in our laboratory, to directly convert one target base to another. And then they also have to guide the cell through DNA repair processes to make this conversion permanent on both DNA strands. Cytosine base editors convert C to T or G to A, while adenine base editors convert A to G or T to C. Nature actually doesn't provide any known enzyme that performs the deoxyadenosine deamination chemistry that's needed to create an adenine base editor. So we evolved in our laboratory the first known deoxyadenosine deaminases in order to develop these adenine base editors. We also developed, on the right, CRISPR-free all-protein base editors that use tail or zinc finger proteins to target DNA, thereby enabling the first purposeful changes in the sequence of mitochondrial DNA in living cells and in animals. It had not been previously possible to make specific changes in the DNA of mitochondria because of the inability of guide RNA sequences, and therefore of CRISPR systems, to functionally enter the mitochondria. Our lab and many others have used base editors both ex vivo and in vivo to treat animal models of human genetic diseases. Shown here are ex vivo base editing to treat sickle cell disease in mice, and at the bottom, in vivo base editing to rescue a humanized mouse model of progeria, in both cases with extensive rescue of disease phenotype. But what about a genetic disease caused by a variety of different mutations, including the deletion of an entire gene? You can't edit, obviously, what's not there. So how can a single gene editing treatment still be developed that might serve all such patients? Well, we recently reported a therapeutic gene editing strategy to treat spinal muscular atrophy, or SMA, which has historically been the leading genetic cause of infant mortality worldwide. SMA is the consequence of two genetic accidents. First, SMA patients have homozygous deletions, or loss of function mutations, in their SMN1 genes, which encode the essential SMN protein. Now, in animals, the loss of SMN1 genes results in a failure to complete embryogenesis. So there's no animal cases of SMA. Humans, however, uniquely have an imperfectly duplicated copy of SMN1 called SMN2, which differs from SMN1 in a single C to T mutation that causes exon 7 to be mostly skipped during splicing. As a result, SMN2 makes a defective, short-lived SMN protein that's insufficient to support motor neuron survival, but it is sufficient to allow SMA babies to be born, only to then suffer from progressive loss of motor function, including the ability to eat and breathe eventually. 3 FDA-approved SMA drugs, including the antisense oligonucleotide nusinersen, have completely transformed the lives of thousands of SMA patients. However, these drugs do not fully restore SMN protein levels, do not allow endogenous regulatory mechanisms to control SMN production, and require repeat administration, or in the case of the AAV gene therapy, Zolgensma, is expected to wane at some point, but it's not clear that we can redose those patients. So we sought to develop gene editing strategies to treat SMA, and rather than try to correct SMN1 mutations, which vary by SMA patient and often are just deletions, instead we targeted SMN2, that imperfectly duplicated SMN1 copy that all SMA patients must have in order to be born. We tested more than 70 nuclease and base editing strategies targeting SMN2 that we thought might increase SMN protein levels. And so shown on this slide are 35 nuclease editing strategies that generate indels that we hypothesized should increase SMN protein production. And the most effective of these 35 nuclease editing strategies indeed increased SMN protein levels by as much as 17-fold. However, SMN protein levels are about 40-fold lower in these SMN cells than in normal cells, so even the best nuclease editing strategies we identified did not fully restore SMN protein levels, in addition to the other downsides of editing through cutting chromosomes. Here are 19 base editing strategies, all of which sought to convert SMN2 into a healthy copy of SMN1 by correcting that C to T mutation in SMN2. And the most effective of these base editing strategies fully restored SMN protein levels. So we packaged the most promising of these adenine base editors, like the D10 strategy, into a dual AAV9 vector and injected ICV neonatal SMA mice lacking SMN1 genes and containing SMN2 genes, human SMN2 genes. And we observed efficient transduction of tissues in the CNS, including 43% transduction of spinal motor neurons. And in the mouse CNS, the chosen base editor efficiently converted that defective SMN2 copy back into a normal SMN1 gene in 87% of transduced cells. We assessed the rescue of motor function by electrophysiology measurements. These electrophysiology assays revealed that the base editor-treated SMA mice showed substantial rescue of motor neuron functional integrity, reaching levels similar to those of healthy mice. Now, we initially tried to treat these mice with the base editor alone and measure their lifespan, hoping that they would show substantially improved lifespan. We did observe modestly increased lifespan, but then we realized that the unusually short time window for treatment in these SMA mice, which is only about maybe 5 or 6 days for correction to take place before the fate of motor neurons is sealed, was likely too short for substantial levels of base editing to occur, which when delivered by an AAV, we know from our other studies, takes typically a week to 3 weeks to take place. Now in human SMA patients, in many respects, it's easier to treat human patients than this mouse model because the window for beneficial treatment is much longer, up to perhaps a year or so. So we hypothesized that simple co-administration of a single dose of nusinersen, the SMA antisense oligonucleotide, together with the base editor AAV, would likely extend the therapeutic window for gene correction in SMA mice, and would also more closely resemble the likely treatment state of SMA patients in a potential clinical trial. And indeed, one-time in vivo co-administration of the base editor and nusinersen greatly extended animal lifespan from a median of 17 days to a median of 77 days, along with substantial rescue in behavioral assays, such as the time required for mice to right themselves after you put them on their back. So this is a video of a righting reflex assay for untreated SMA mice at 14 days old. And you can see that these mice struggle to right themselves if you lay them on their backs. In contrast, here are videos of the SMA mice that received a single injection of the adenine base editor. These are also at 14 days old, and they show substantial rescue of writing ability. So together, these data suggest the potential of a one-time base editing treatment for SMA, and we're trying to advance these studies into IND-enabling studies. The beautiful work of scientists in other academic labs and in industry have advanced base editing into primates and into clinical trials now. So Kiran Misunuru's lab and scientists at Verve Therapeutics and Beam Therapeutics recently reported the one-time LNP-mediated delivery of adenine base editor mRNA into the liver of non-human primates, resulting in precise mutation of a PCSK9 splice site. Even though their goal was to simply lower PCSK9 levels, and therefore in principle they could use a nuclease to knock out PCSK9, they actually compared side by side Cas9 nuclease with base editors to achieve this goal, and they chose base editors due to their observation of both higher efficacy and fewer unwanted outcomes. And so LNP-mediated adenine base editor delivery durably reduced PCSK9 levels in monkeys by 90% and LDL cholesterol levels by 60%. There are currently at least 6 base editing clinical trials underway. In late 2021, in the top row of that table, FDA granted IND clearance for Beam 101. This is the first US base editing clinical trial, which uses an adenine base editor to install naturally occurring mutations in fetal hemoglobin genes to treat sickle cell disease. In mid-2022, in the second row of the table, Wasim Qasim's team in the UK treated the first patient in a base editing clinical trial, which used multiplexed base-edited CAR T cells to treat T-cell leukemia. In late 2022, the first patients were dosed in an in vivo base editing clinical trial. That's Verve 101 that precisely installed those splice mutations in PCSK9 to treat familial hypercholesterolemia. Last month, Beam announced the first dosing of a patient in the US with a base editing therapeutic, Beam 201, for ALL and AML. And at the end of last year, the first clinical outcome from a base edited therapeutic was announced. This is Alyssa. At the time this photo was taken, she was a 13-year-old girl in the UK with T-cell leukemia. And she was given a very poor prognosis after both chemotherapy and a bone marrow transplant failed to clear her cancer. She was infused with CAR T cells containing 3 base edits in Wasim Qasim's trial, and following treatment with these triply base-edited CAR T cells, her T-cell leukemia went into complete remission, and as of last month, which is about 17 months after treatment, her cancer remains undetected. As you can read in this letter that her family sent to our lab last month, Alyssa is now back in school and hopes to pursue a career in biomedicine. So the fact that it only took 6 years to go from our first base editing paper in 2016 to the first positive clinical readout is really a testament to the work of thousands of laboratories around the world who have collectively published more than 1,000 papers that include base editing to date. So base editors can make targeted transition mutations in cells, animals, and patients, but what about pathogenic mutations other than the 4 transition mutations? These other mutations account for about 70% of known pathogenic gene variants. So we sought to develop other ways of installing or correcting transversions or deletions or insertions, again, without requiring double-strand DNA breaks or donor DNA templates. Now, the first 2 base editors each took about 2 years to develop. And so initially we thought, well, it would probably take the field another 10 years or so to develop all of the remaining molecular machines needed to complete the rest of this pie chart. But in 2018, a very talented former postdoc in the lab, Andrew Anzalone, initiated an effort to develop a new way to do gene editing called prime editing. Prime editors are fusions of programmable nicases with engineered reverse transcriptases. They use an engineered prime editing guide RNA, shown in green here, or PEG RNA, as we call it, shown in green, that not only specifies the target site like a regular guide RNA, but it also encodes the desired edit. Prime editors nick the target DNA site, and then they use the 3' end of the freshly nicked DNA strand to prime the reverse transcription of an extension on the PEG RNA that serves as the reverse transcriptase template. The engineered reverse transcriptase domain then copies the desired edit directly onto the end of the target DNA strand, creating a 3' flap that the cell eventually resolves into a heteroduplex containing one edited strand and one edited strand. And the magic of this resolution largely has to do with the fact that 5' flaps are very common intermediates in DNA damage and DNA repair, repair and DNA replication. And therefore, cells have mechanisms to efficiently cut off 5' flaps and ligate them. And so eventually these 3' flaps end up becoming incorporated into DNA. Now, the most efficient prime editing systems nick the non-edited strand, the bottom strand in this cartoon, to stimulate the cell to remake that strand using the edited strand as a template, which completes the permanent editing of both DNA strands. Now, the whole point of editing DNA this way is that the PEG RNA's reverse transcriptase template is specified entirely by you. So you can make virtually any kind of substitution, deletion, or insertion using this strategy. And indeed, that's the hallmark of prime editing, this remarkable versatility. You can now perform all 12 possible types of DNA substitutions, as well as precise targeted deletions and precise insertions for the first time in mammalian cells without requiring double-strand DNA breaks or donor DNA templates. More than 95% of known pathogenic mutations are smaller than about 50 base pairs in length, and thus are well within the size range for installation or correction with prime editing, which in its current form can mediate deletions of up to maybe 1,000 base pairs and insertions of up to at least a couple hundred base pairs in using simple prime editing. Our lab and several others have reported the use of 2 PEG RNAs to simultaneously install both edited DNA strands. Doing this obviates the need for the cell to perform any kind of second strand synthesis. We call this method twin PE, and it's an especially efficient way to perform prime editing of larger sequences, such as the insertion of these @P or @B recombinase landing sites for BXB1 recombinase. Twin PE combined with a recombinase can then be used either in a 2-step process or in a single transfection process to knock in large gene-sized DNA at target sites of our choosing in the human genome, including at safe harbor loci or at the endogenous sites of pathogenic gene loss. And so these efforts represent the first RNA-programmed site-specific insertion of gene-sized DNA in mammalian cells, and we're hopeful that this kind of strategy might help address one of the persistent challenges in our field, which is for certain genetic diseases, there are hundreds of different mutations that cause the disease, and it seems impractical to imagine developing a different drug to treat each one of those mutations. So the hope is for certain loss of function diseases, perhaps you can treat them by inserting a healthy copy of the target gene at the endogenous locus, where it will be subjected to endogenous regulatory mechanisms. And Jonathan Gutenberg and Omar Abudaya have described a similar method that they call PASTE that also uses prime editing to install recombinase sites, and I believe you'll hear from Omar in this conference on Tuesday. Previous CRISPR-based editing methods derive their DNA specificity primarily from a single DNA guide RNA hybridization event. In contrast, prime editing requires 3 such hybridization events to take place in order for prime editing to take place productively, And each of these hybridization events presents an opportunity to reject an off-target sequence. So we wondered whether, as a result of these sort of 3 checks that all have to be passed, whether prime editing might be inherently mechanistically resistant to off-target editing more than, for example, a simple CRISPR nuclease. So to test this possibility, we examined 16 known off-target sites in the human genome of 4 previously characterized guide RNAs. And we compared side by side the editing efficiencies of Cas9 nuclease versus prime editors at each of these 16 off-target sites using the same PEG RNAs to program both editing agents. And we found that prime editing was indeed much less common at these off-target sites than Cas9 nuclease-mediated editing. And many other labs in reports cited at the bottom of the slide have also investigated off-target prime editing, alterations of telomeres, changes to endogenous genomic retrotransposons, altered splicing patterns, altered gene expression patterns, using a variety of methods, including whole genome analysis and whole transcriptome analysis. And thus far, these studies have found prime editing to be highly specific for the target sequence, with no observations yet reported of Cas-independent, sort of targeted independent prime editing consequences. Another important aspect of prime editing is that the edited products tend to be quite homogeneous. So here's an example shown in 2 different human cell types. The goal of this example is to insert this 3-base insertion of AAG into a certain site in the human genome. And you can see that comparing the unedited cells at the top with the edited cells, where this slide shows every high-throughput sequencing read that was observed at a frequency of 0.2% or more, that the vast majority of the edited DNA, when you treat with a prime editor, has the desired correction. In contrast, if you treat with a Cas9 nuclease and an HDR template to install the same 3-base pair insertion, you get mostly a large mixture of these different indel byproducts, even in cell types like K562 cells that support reasonably efficient HDR. All right, over the past few years, we've sought to improve prime editing systems in a variety of ways. We started with achieving a deeper understanding of the cellular determinants of prime editing outcomes. In collaboration with Britt Adamson's lab at Princeton and Dr. Jeff Hussman and Jonathan Weissman's group, we used their so-called Repair-seq CRISPR-I screening platform to knock down several hundred different DNA repair genes excuse me, in human cells. And then we measured quantitatively how each of these perturbations affected prime editing outcomes. The strongest single family of hits from the screen were all components of the DNA mismatch repair pathway. And so if you knock down these genes in blue, you strongly increase prime editing efficiencies, and you also decrease the frequency of indel byproducts, consistent with the mechanism of prime editing I showed earlier. All right, so with that knowledge, how can we improve prime editing systems? How can we evade mismatch repair during prime editing? Well, in mammalian cells, mismatch repair primarily operates on heteroduplexes containing 1 or 2 mismatches. If you have more than 1 or 2 mismatches, the resulting heteroduplex is a poor substrate for mismatch repair. So we exploited this fact to evade mismatch repair during prime editing by carefully designing our prime edits to purposefully install additional benign or silent mutations, represented in blue here, near the target edit so that those prime editing intermediates natively evade mismatch repair. And indeed, we observed that simply adding additional silent or benign mutations near the target site can substantially improve prime editing efficiencies. We now routinely use this strategy to design all of our PEG RNAs. So this is a counterintuitive advance. You wouldn't normally think that editing extra things makes your edit easier rather than harder, but it came entirely from achieving a deeper basic science understanding of prime editing and how the cell responds to it. We also optimized codon usage, Cas9 sequences, linker sequences, nuclear localization sequences to generate an improved prime editing protein architecture that we call PEMAX. And in work I don't have time to present in detail, we discovered that capping the critical 3' extension of pegRNAs with certain RNA pseudoknots yields what we call ePEG RNAs that resist cellular degradation, probably something that this community could have told me in advance, which really enhances prime editing efficiencies, especially in vivo. And when we combine these three improvements, mismatch repair evasion, an improved protein architecture for the prime editor protein, and ePEG RNAs, we observe a large cumulative benefit consistent with their independent mechanisms, such that prime editors with ePEG RNAs that evade mismatch repair and use the optimized architecture outperform our original prime editing systems on average by more than an order of magnitude in both editing efficiency and in ratios of edits to undesired indels. These advances in prime editing systems have had a profound impact already on our ability to make challenging therapeutic edits, especially in mismatch repair competent cells that used to be poorly edited. So here are just 3 examples of projects that are in progress, all of which began with low single-digit % prime editing, but now through these successive insights can all now be edited at useful levels. The ability of prime editing to precisely replace one stretch of DNA of fairly large size with another stretch raises the possibility of correcting the root cause of trinucleotide repeat disorders. And so in some exciting recent data, the enhanced prime editing systems I described in this talk can precisely replace larger numbers of GAA repeats at the frataxin locus, which is the cause of Friedreich's ataxia, with non-pathogenic smaller numbers of repeats efficiently and with very few indel byproducts. Scientists at Prime Medicine have applied a similar approach to cells from Friedreich's ataxia patients, resulting in the restoration of frataxin protein levels in patient-derived iPSCs and the rescue of axonal projections in dorsal root ganglia that were differentiated from these patient iPSCs. We've also used prime editing to replace directly a larger number of CAG repeats at the HTT locus, which is the cause of Huntington's disease, again with a non-pathogenic smaller numbers of repeats in cells from animal models of Huntington's disease and in mouse models. Researchers have also, just like with base editing, now used prime editing to treat animal models of genetic disease, both ex vivo and in vivo. We recently used ex vivo prime editing of sickle cell disease patient hematopoietic stem cells to efficiently correct the sickle cell allele, this time back to wild-type beta globin, because we can use prime editing to make a T to A change, something we can't do with a base editor. And we rescued sickle cell disease phenotypes after you transplant the resulting hematopoietic stem cells back into mice. So 17 weeks after transplantation, the prime-edited hematopoietic stem cells show engraftment frequencies, hematopoietic differentiation, and lineage maturation that's indistinguishable from those of unedited hematopoietic stem cells, suggesting that prime editing didn't impair hematopoietic stem cell engraftment or differentiation. And an average of 42% of human red blood cells isolated after transplantation of the prime-edited cells from these sickle cell patient donors into mice express wild-type beta globin, exceeding the threshold of about 20% thought to confer therapeutic benefit. The red blood cells from transplanted mice carry less sickle globin if they receive the edited cells and resisted hypoxia-induced sickling. And so these findings just suggest the feasibility of a one-time prime editing treatment that offers a fairly unique combination of reverting the pathogenic allele back to the wild-type allele without requiring any viral DNA or really any DNA at all. There's no DNA in this treatment, and also minimizing the undesired consequences of making double-stranded breaks. Researchers have recently described in vivo prime editing using a variety of viral and non-viral in vivo delivery methods. For example, in work we recently reported, our lab achieved in vivo prime editing using engineered split AAVs, resulting in therapeutically relevant levels of prime editing in the mouse brain or liver, as well as some significant prime editing in the bulk mouse heart. And at this meeting on Tuesday, Seth Alexander from Prime Medicine will present some really exciting non-viral prime editor delivery data using LMPs to deliver mRNA encoding prime editors into mice to treat glycogen storage disease type 1b, GSD1b. Recent papers have also described in vivo prime editing in mouse models of human genetic diseases. For example, Kai Yao's lab recently reported the use of AAV-delivered prime editing to rescue retinal degeneration in a mouse model of retinitis pigmentosa, and Gerald Schwank's lab used adenoviral delivery of prime editors to rescue phenylketonuria in mice. All right, so the last scientific story I wanted to tell you is how we actually develop and advance some of the editing agents themselves. I gave a talk recently at an event with the legendary Dean Kamen, the founder of Segway and the founder of the first programs that have gotten many kids interested in science and technology. And Dean Kamen said he's always wanted to know how the magic goo is made. That's what he calls these molecular machines that people in the audience work with. And so this is a story about how the magic goo is made. So despite all of this progress on prime editing, the reverse transcriptase that's at the heart of the prime editor machine has proven to be the most challenging component to improve, despite the efforts of many labs. And the remarkable ability of the original PE2 prime editor to perform prime editing became obvious when we tested 59 natural reverse transcriptases from 14 different classes and identified that 20 natural reverse transcriptases demonstrate detectable prime editing activity, but none of them are as active as our original PE2 prime editor, especially for longer or more complex edits like the ones shown on the right. So in work we just reported, we applied our phage-assisted continuous evolution, or PACE platform, to generate highly evolved prime editor proteins with greatly improved editing properties. So PACE is a way that we accelerate laboratory evolution of proteins and, if you wish, RNAs to speeds that are much, much faster than our slow primate hands allow us to otherwise evolve genes. So during PACE, phage-carrying gene variants encoding proteins with the desired activity replicate despite being continuously diluted, while phage-containing genes lacking the desired properties are washed out of this fixed volume vessel that we call the lagoon. Since the phage life cycle can take place as quickly as every 10 minutes, PACE enables evolution campaigns that are literally hundreds or thousands of generations of mutation, selection, and replication, what used to require hundreds or thousands of weeks to accomplish, to now take place on a practical timescale of, say, days to small numbers of weeks. So PACE of prime editors requires linking prime editing to phage propagation. And one of the genetic circuits we use to establish this linkage is shown here. In this case, prime editing of an inactivating mutation in T7 RNA polymerase restores T7-mediated expression of gene 3, which is an essential phage gene, which enables phage propagation. So that's the circuit. And one of the first findings when we began to use this circuit to evolve prime editors is that we realized prime editors were evolving in different ways in response to being required to perform different types of prime edits. And that's actually different than the prevailing view at the time and different than the situation with nucleases or base editors. So the selection circuit in yellow requires a one base pair insertion to fix T7 RNA polymerase. And the prime editors that evolve from this selection, shown in the yellow bars, are particularly good at making this one base pair insertion. But if we use a selection circuit in blue that requires a 20 base pair insertion, the evolved prime editors that come out are good at making the 20 base pair insertion, but aren't particularly good at making the one base pair insertion. So this experiment revealed that the optimal prime editor for a given type of edit depends on the nature of that edit for reasons that we figured out, and I'll explain in a few slides. Right, so we applied PE pace, as we call it, to the most promising of the wild-type reverse transcriptases, starting with compact reverse transcriptases that are much smaller than the engineered MMLV reverse transcriptase in PE2. Smaller is good because smaller means that they're easier to deliver using size-constrained delivery methods like AAV. And in all cases, PE pace rapidly evolved new reverse transcriptase variants with greatly improved prime editing capabilities in mammalian cells, which you can see here comparing the colored bars representing the evolved variants with the gray bars that use prime editors with wild-type reverse transcriptases. When we tested the best of these pace-evolved compact prime editors on a variety of edits in human cells with that of PE max, the previous gold standard, we were happy to find PE6A and PE6B offered performance comparable to that of PE max, but were about half the size of PE max. Now, in an effort to create prime editors that improve PE max's ability to perform long, complex prime edits while maintaining a smaller size, we pursued multiple different approaches in parallel. In the first approach, we extensively tested rationally designed mutations in TF1 reverse transcriptase designed to enhance binding to DNA or RNA substrates as potential strategies to increase the processivity of the resulting reverse transcriptases. And we combined the most helpful of these mutations to pace-evolved PE6B, and that resulted in PE6C. In the second approach, we subjected PE2, our original prime editor, to pace using 3 different selections, including 2 that required this 20 base pair insertion for survival, this more challenging prime edit. We originally thought that we would have to repeat this study with truncated versions of PE2 in which we deleted the RNase H domain, which is thought to be not essential for prime editing. But as is often the case, PACE was smarter than we are, because when we subjected this gene to PACE, it naturally told us that you don't need this RNase H domain because it evolved premature stop codons and frame shifts in the RNase H domain that naturally truncated that part of the gene off. And so in addition to other mutations, and when we tested and combined the resulting mutations, we ended up with PE6D, which you can think of as a PE2 descendant that lacks its RNase H domain, yet has been evolved for high performance on long prime edits, something that PE2 missing its RNase H domain typically performs poorly at. So PE6C and PE6D perform particularly well on these long prime edits. But when we started to characterize their properties, we began to realize that their benefits were not simply a matter of the length of the prime edit. So here's an example. There's 2 prime edits shown on this slide, both fairly long insertions. And you can see in the top case, PE6D does much better than truncated PE max. In the bottom case, however, both prime editors perform quite similarly, even though both edits require this long reverse transcriptase template. But we noticed that the top edit has a reverse transcriptase template that's predicted to form a giant hairpin in which the reverse transcriptase has to plow through that hairpin in order to complete the prime edit, while the bottom edit lacks the hairpin. And so when we performed a terminal transferase tagging and pull-down experiment, which captures all of the prime editing intermediates mid-edit in a cell, we observed that indeed the PE max prime editor stalled frequently at that hairpin in the upper case, so that only about half of the prime editing intermediates reached all the way to the end of the prime edit, the reverse transcriptase template. In contrast, PE6D had no problem plowing through that hairpin such that the vast majority of the prime editing intermediates reached the full end of the reverse transcriptase template. In another telling experiment, when we purposefully designed a reverse transcriptase template to fold into a giant dumbbell, we observed, again, that PE6D performed much better than PE max. But if you introduce mutations that break up that secondary structure, now both prime editors perform comparably. So these and other experiments led us to realize that it's the folding energy of the reverse transcriptase template and primer binding site that dictate which type of prime editor you should use to make different kinds of edits. And once the predicted secondary structure of the reverse transcriptase template is more stable than about minus 23 kcals per mole, that's when you should use PE6C or PE6D to make the prime edit. So you should not use PE6D or PE6C for simple substitutions or when using unstructured reverse transcriptase templates, because their high processivity increases the frequency of reading into the guide RNA scaffold, which results in indel formation. So that's why there isn't one prime editor that's ideal for all different kinds of prime edits. The benefits of these PE6s are particularly pronounced for more challenging edits or in more challenging cell types. So here are some disease correction edits that are more complex than simple substitutions, including in patient-derived cells, and you can see that the PE6 variants substantially outperform our PE max prime editor in all these cases. And consistent with this trend of the PE6 prime editors performing particularly well in the most challenging settings, we observe the strongest advantages of the PE6 variants in vivo. So following the ICV injection into the brain of dual AAV prime editing systems, we observed that PE max performed fairly poor dual flap insertion edits, such as the insertion of recombinase landing sites into a safe harbor locus in the mouse brain. But in contrast, PE6D yielded order of magnitude improvement in editing efficiency. And when we did the same types of experiments in the mouse brain using single flap large insertions, we again observed very large improvements. If you can't see it because of the podium, I apologize. Although I'll point out that whoever advertised here is getting the best deal for their dollar ever. But we see more than order of magnitude improvements in editing efficiency when we use PE6D over PE max, again, in mouse brain for these single flap edits. So we've released a lot of editing agents to the public. We open source everything so that everybody can use these technologies and improve on them and characterize them. And frequent feedback we get is, you release so many different editing agents, I don't know which one to use. So here is the answer key for which prime editor variant you should use to make which prime edit. Basically, we suggest that you start with the size requirements of the prime editor application. If you need the prime editor to be as small as possible, PE6A is the way to go because it offers PE max-like editing efficiencies, but it's much, much smaller. When you have to fit prime editors into dual AAV systems for in vivo delivery, we recommend testing PE6B, 6C, and truncated PE max for edits using weakly structured reverse transcriptase templates, and testing PE6C and 6D for edits requiring highly structured RTTs. And if you don't have any size constraints, the same recommendations apply, except you can also now add full-length PE max, which does not fit into two AAVs, to edit using for edits using highly structured reverse transcriptase templates. All right, so as editing technologies continue to advance, one of the most important challenges facing the field is how can we bring the promise of these editing technologies to as many patients as possible? Now, one small answer may lie in the mechanism of prime editing, because a single prime editor plus a single PEG RNA, which qualifies by FDA standards as one composition of matter, can correct many different pathogenic alleles, since a single prime editor can actually pave over a whole stretch of nucleotides of, say, 100 or even 200 base pairs that's separate from the DNA sequence that's recognized by the prime editor. So this aspect of prime editing may help address this long-standing challenge in therapeutic gene editing that arises from the fact that, unlike sickle cell disease, unlike progeria, where the genetic disease is all caused pretty much by one mutation, most genetic diseases are not caused by a single mutation. And so here is an example. This is CDKL5, which, as many of you know, causes mutations in CDKL5 cause CDKL5 deficiency disorder. And there are hotspots of many different pathogenic mutations, and you can use a single prime editor to pave over the entire hotspot, thereby installing or replacing any or all of those mutations at the same time. But for many genetic diseases, the mutations that cause the disease are not clustered. like Stargardt's disease, like Batten's disease, like many Rett syndrome, many of the diseases you guys work on. For these diseases, the advent of versatile and precise gene editing technologies that can correct a large fraction of known pathogenic mutations simply by changing one guide RNA now creates a sort of hurry up and wait situation that we all have to work at to resolve. The situation is, how can we best serve the broadest set of genetically diverse patients with these new capabilities so that developing new treatments for 100 pathogenic mutations in one gene doesn't consume 100 times the amount of time and resources that it took to develop the first treatment. So some potential answers to this critical question might include, could one animal model for a genetic disease support clinical studies for multiple gene editing treatments that correct different mutations in that gene, rather than requiring a different animal model for each mutation in the same gene? Could one set of in vivo tox studies for the combination of an editing agent and a delivery method de-risk the use of that combination to correct multiple different mutations in the same gene? And if so, if you just change the guide RNA to correct a mutation a little bit downstream or a little bit upstream of the first example, can we de-risk subsequent guide RNAs using off-target experiments that are performed in cultured human cells rather than in animals? And finally, providing incentives to pursue the development of gene editing drugs to treat rare genetic diseases will be critical to providing access to many of the patients in greatest need. And so an idea that was shared, I think, by some members of this community, including Tim Yu, perhaps priority review vouchers, which, as you may know, can be auctioned for tens of millions of dollars to pharmaceutical companies, can be offered by FDA to organizations upon first dosing of patients in rare disease clinical trials, rather than upon first drug approval. Okay, so to wrap up, here's a summary of the three classes of programmable gene editing technologies that function robustly in mammalian cells. Nucleases can efficiently disrupt target sequences, but they generate uncontrolled mixtures of indel products, and they induce undesired consequences of chromosomal cleavage. Base editors efficiently perform the 4 types of transition mutations without requiring double-stranded breaks, but they're vulnerable to bystander editing when a non-target A or C base is located right next to a target base. And older base editors can result in Cas-independent deamination of DNA or RNA. Prime editors can make virtually any kind of local edit, again, without requiring double-stranded breaks and with high DNA specificity, and with minimal possibility of bystander editing or Cas-independent editing due to its mechanism. But prime editors are newer and larger than nucleases or base editors, and typically require testing of many PEG RNAs in order to achieve high editing efficiencies. All three of these technologies have now rescued many animal models of human genetic disease, and the first 2 have entered patients in multiple clinical trials, with the first prime editing INDs anticipated to be cleared next year. I'll end with just a few thoughts on what the future of therapeutic gene editing might look like, classified by their historic rate of change. So perhaps contrary to recent popular perception, I actually think fundamentally distinct gene editing technologies are slow to arrive, or at least unpredictable in when they will arrive. It's been 70 years since we've known the molecular structure of DNA, and yet we only have 3 fundamentally distinct ways to edit target DNA in human cells that are efficient, specific, and occur at target sites of our choosing. On a more optimistic note, I think the field has repeatedly demonstrated that once conceived, nucleases, base editors, and prime editors have all been rapidly improved and diversified to enhance their editing efficiency, to enhance their specificity, and to streamline their ability to be delivered in vivo. So I wouldn't call these important improvements easy to accomplish, but I think they're easy to anticipate. And so in my opinion, that leaves these two components of the therapeutic gene editing enterprise that are likely to be rate determining given the above. In vivo delivery technologies, and there's a whole session in this meeting devoted to in vivo delivery technologies, as well as regulatory processes. A heart surgeon recently pointed out to me that he can perform experimental life or death heart surgery on a child in the US, and he only needs institutional sign-off by his local IRB committee. But experimental genome surgery on a patient gravely affected by a genetic disease requires a multi-year, sometimes 10-year approval process. So I hope we can all work together to help regulatory agencies develop processes for these technologies and for the treatment of rare genetic diseases in general with the same degree of innovativeness and creativity that's driven so much of this science forward. All right, as this molecular rendering of prime editing from David Belinsky's amazing team plays in the background, I'll just end by noting that not that many years ago, the concept of using laboratory engineered and evolved molecular machines to find and correct a specific mutation in an animal or a human patient to alleviate the consequences of a genetic disease really seemed kind of crazy. So every morning I wake myself up and pinch myself that I have the opportunity to work with such an incredibly talented group of graduate students and postdocs, and especially these collaborators who have made all of this work possible. Thank you so much for your attention, and I'm happy to answer any questions.
Thank you very much, David.